in-depth discussion for the half hour, and I just wondered whether they'd solved it within that half hour. Because it may be the first time the three of them have got together in that sort of context, who knows.
It is.
It is, fine, good. No questions then. So what we'll do is the person who has the microphone has the voice, okay, and then I'll try and control people. Who'd like the first question? Rachel, off you go. If you could just state the name perhaps.
Yeah, I'm Rachel Warby. I've been emailing you, and I'm the dripping tap I've been called, as annoying as a dripping tap. Due to my own personal experience, after my boxer had Novivac L4 in 2014, I then noticed similarities with other boxers becoming seriously ill and dying young of renal failure. They had all been administered the vaccine. I have been busy collecting and researching evidence myself and found other like-minded owners and breeders that have heard of cases of adverse reactions, including JKD-like renal failures. There is evidence of the cases in other breeds that are extremely similar to boxers. If these dogs were boxers, they would stand out as potential JKD cases, which couldn't be right if it was just breed specific. So there needed to be another cause. The one I suspect is the lepto vaccine. The gene is really important regardless of the influence of the vaccine. However, if using the vaccine is causing premature induced renal issues in dogs with a possible form of JKD, were these pups or dogs not to have the lepto vaccine, maybe they would have a fuller life with less or no symptoms. If it could be the lepto vaccine that's causing an unnecessary suffering and premature death, it could also be causing issues and obstacles locating the gene due to mimicking the disease in some dogs that don't actually have it. We need to rule it out and also separate the cases from hereditary JKD only, vaccine only, or vaccine triggered premature JKD. I have used the information collected and I've invested my own money to have a professional vet from America, Patricia Jordan, who's helped me with my own personal research. And I just wanted to ask, it seems that there are less cases coming from breeders, and maybe that's because as breeders we now realise the L4 has been problematic. And it seems now that the pet owners are the only ones that are still kind of using the L4 vaccine because their vets are recommending it. So what I would like to put forward is that there was a problem with the L4 vaccine, that we're not now using it, so our cases are not coming through as much. And we need to rule out the vaccine.
So maybe I'm best placed to respond to that particularly. So firstly, I'm very sorry to hear about your personal dog that you lost. So as a specialist in internal medicine and particularly in the kidney, I have to say I know of absolutely no evidence that leptospirosis vaccination causes kidney disease. I know that if you look on the internet that there are people that are saying there are, but I can say that there are people that are saying vaccines cause almost every disease that happens in dogs and some in cats. And just like with the situation with the MMR vaccination, it's very difficult if you look for any type of disease and then you say, well, was it vaccinated? Was it vaccinated more recently? Actually, it's very common to come up with an association, particularly with owners who may have, they may be more likely to have gone to the vets because, well, they've saved up going to the vet because they know that they're going for vaccination, and then when they take the patient for vaccination they say, oh, and actually he's doing this or she's doing this or there's this problem, and then the investigation starts at that point. And so it's very easy for there to be an association made with vaccination. So I personally, I vaccinate my own dog for lepto, i.e., well, when he's going into kennels. I'm not very good at vaccinating her generally, but the reason that I don't do it is not because of any concerns with vaccination being associated with any disease. So I know of no reason to suppose that they are associated. I would also say that actually, if you're trying to implicate the L4 vaccine specifically, then actually the papers that we wrote, like the one Marge Chandler and I wrote, that series of dogs was before L4 was in the market in the UK. So we were already seeing a problem. At that time, people were only using the L2 vaccine. There is no evidence for a link, and you'll find there are lots of vets out there who'll say lots of things. I mean, I think most of the stuff in terms of the anti-vaccination has been associated with the sort of raw feeding, the sort of not always the same thing, not always raw feeding, not always homeopathy, but a lot of these things are put together in the same camp. And yeah, I just don't buy it, I'm sorry.
I disagree. I've got the evidence to show that it does.
Right, Fraser. The question I would ask is, when did JKD come to light? Was the reason that in the past we had 7 or 8 kennels of wildly different breeding, which Bruce will remember as I do? Or is it that it came to light due to advances in veterinary medicine?
Question, yeah. Who's going to do that?
Well, I could say something on that, because we have JKD going back at least as far as 1980, well established in indicators coming up thereafter. The problem with Rachel's discussion, as far as I'm concerned, on vaccines is not only is vaccines just coming recently, but also our problem in this country is just about all our boxers have the same pedigree. So it's very hard to make a case one way or the other as far as the genetics level. But the key for me is the genetics. If they're coming in the same family, then it's likely to be the same thing. It's not likely to be as sensitive to L4 or whatever. You're getting JKD all over the world, which is, it just doesn't fit.
If I can add to that from the veterinary perspective, I think actually vets in general don't particularly know of or recognise a problem in boxers per se, and although I'm a kidney nerd, a dog kidney nerd, and I know of genetic diseases in particular breeds that, you know, English Bull Terriers get polycystic kidney disease. It was described in some of them in Australia, but it's a tiny number of dogs, and I know about it because it was well documented and it was described. But your vet who sees dogs and cats, you know, just in general practice, there's no way that they're going to retain that information. I can do that because I particularly focus on the kidney, and I don't deal with heart disease, and I don't have to do spays, and I don't do all those other things. But in terms of actually finding these diseases, it's not that vets are now doing some newfangled test that is bringing it to light. It is true though that I think vets are more likely to actually do blood work and to do tests, and so it may be identifying that there is a kidney problem, but it's not identifying which kidney problem. dogs have.
It was just when was it brought to light? Was
Yeah.
it?
So I guess
and why?
well, the first descriptions of a boxer disease were done by pathologists, and they were basically just working in the vet schools, looking at patients that arrived in the post-mortem room, and then when they looked at their kidneys, some of them had particular changes, or they were a young dog, and so they just wrote a description of that, and that was how the first descriptions occurred. And boxers weren't alone in that. There's actually a long list of breeds that have sporadically had these diseases recognised.
Question from someone?
It's just more on a positive note of something we've picked up today, and we've sat in a break, like going all through our pedigrees. We look at the kennel club coefficient, like for the mating we want to do now, and you get a low number, but then you look at the pedigrees and you start to see the things where you are inbreeding back. So what we've sat doing today, and I think it was the professor who brought it up, let's look at the coefficient of our breeding in the past. And we've sat here today doing it, and we're all like, but that's really low. So the back of us, we've got really low that maybe we can actually risk going a little bit closer now that we're all backing away from. If when you look back at your lines, you can see that matings we've done are coming so low in the past that we're not looking at our pedigrees with these coefficients thinking the older the pedigrees are, when it's not. It's what the kennel club are saying to us, that's 16% or that's 18. You're going ahead and doing it, but then when you look at the pedigrees, they're still quite risky. If we've gone back to parents, grandparents and great-grandparents just sitting here today, seeing that they're coming in so low, and that was what you said to do, so we're not looking at our pedigrees going, oh, you know, with fear now. We've seen that over the years, we've not inbred that tightly, and we have got lower coefficients going right back. So I'd like to say I've took that as a light bulb moment from you today, that we can actually look back at the coefficients and see what risk we're at. So
Yeah,
I found that useful.
So that's a really good point. I'd say that inbreeding is massively misunderstood. People seem to take it as a completely static phenomenon. In fact, there are things called naked mole rats, which live underground in little colonies, and because they are, well, people describe them as penises with teeth. They're just these sort of wrinkly pink things with teeth at the front end, and they can't go out on the surface to go out and find other mole rats, so they basically mate with their close siblings. They're like bees. They have a queen, and then she mates with all her male relatives. They're fantastically inbred, but they're fine because they've been doing it for eons and eons and eons. And actually, inbreeding is all right as long as you've got rid of all the genetic rubbish. The problem is when you go from a big population, like mongrel dogs, and you decide, right, we're going to have a breed that looks like this. And the initial stages are going to be worst because you're going to get high coefficients of inbreeding and quite a lot of genetic rubbish to get rid of. But all the sick dogs take with them, who die early, take with them some of that genetic rubbish. So it does become cleaner as time goes by. So the risk of inbreeding depression, illness due to inbreeding, actually changes over time as the genetic load changes. So actually some of the breeds may be, certainly the inbreeding risk, if you did that in humans or a species with a large population, would have much worse consequences than it does in pedigree dogs. However, there are a few cases where you haven't got rid of the rubbish, you've just fixed it. And so that's things like, I don't know, deafness in Dalmatians and things like that, where they've gone to high frequency. I think there are some cancer risk genes in Scotties I read about, and there are various things which have gone to high frequency and haven't been got rid of, and there the inbreeding is still a problem. So unless you understand the genetics in really fine detail, the crude, this is an inbreeding coefficient of this, doesn't tell you an awful lot, because you get an awful lot healthy boxers who have quite a high as I've seen, quite a high inbreeding coefficient. But that doesn't mean it's safe to go that high.
Yeah.
It's usually better to go lower than higher.
No, but
But it's not a golden rule. You can't
Yeah.
really say, right, you're above 10%, you're stuffed.
that is what's happening with the coefficient, isn't it?
Yeah. So the golden rule is always keep lower,
Yeah.
but it's often not as bad. I think the outcome of people who do breed more intensely, as the past has shown, because I think the breeding in recent past was much more intense than it has been recently. So actually, you're probably in a pretty golden period, because as soon as you get less inbreeding, actually the problems will be considerably less. So you're almost benefiting from probably a lot of mortality and unreported mortality that went on when the breed was, when there was much more intense inbreeding. So it really isn't as simple as it's often portrayed, and in a way the history of the breed has as much influence as what you're currently doing on what the outcome of any breed, any given mating will be.
This is still the same topic.
Yeah, Bill Lambert, the Kennel Club. I have bull terriers which also have, I would say, juvenile kidney disease, because we have a population that do die young. A word, just some bad news for everybody here really, that we've known about kidney failure in bull terriers for certainly 40 years, and we still seem to be no closer to finding a genetic solution. But to come back to the inbreeding question, really just to back up what you've just said, first of all, a word of caution. The Kennel Club gives a nice useful inbreeding coefficient for your breed, but actually it's not quite as simple as that, because that figure is really an indication. Once a dog first hits our database, it gets allocated an inbreeding coefficient of 0 if we have no history, which actually can give a slightly misleading result. That dog that hits the database could actually be half brother, half sister, and yet it will give an inbreeding coefficient of 0 because we have no history. So you have to be a little bit cautious. The other thing to remember is you can have two inbred dogs. If they're not related, then the progeny will actually be lower than the parents and less risk. But that's just a word of caution. It's a useful tool, but it's no more than that. I've actually got a question for Bruce. If you could, Bruce, you talked about ratios, about dogs in the UK, you gave a ratio. I didn't quite understand that. Could you give a little bit more explanation to that? You compared ratio with dogs in the UK and dogs in Sweden, etc.
Talking about the sex difference.
No, you gave a popular way. It may have been that's what I didn't understand.
It was sex differences.
Okay, so the sex difference in the UK was different than it was in Sweden and
Well, it's very similar, apart from the Norwegian one, which more males seem to be affected at later ages, which is interesting.
Okay.
It's saying, it's not like some of them are getting through and surviving, breeding them. That's a great word. But on the inbreeding thing, I could just put a point. I mean, I was brought up in an era when it said that every one of us are carrying seven nasty genes, reason why human populations don't inbreed, etc. In dogs, I didn't believe that for a minute, because so many of us way back then did all sorts of breeding, and you didn't get things churning out. It must have been a great sieving over the history of the breed, or the history of dogs, to get rid of so much. On the other hand, a couple of other interesting things. We mentioned the mole rats. They survive. They survive because they're very strong selection for surviving. They don't continue on and breed if they've got something wrong with them. These populations start from almost 0, total wipeout. I think the red kite might be almost in this category. They are producing mad. But birds particularly, I think it's only about 10% survival from one year to the next, young one generation to the next. There's a lot of selection, which we don't do in dogs. So, Faulkner, my professor back in genetics, he started numbers of inbred lines of mice. His figure was you had to start with about 20 before you could make an inbred line. That's brother-sister mating. 2 or 3 generations really going downhill, and then some survive. And then they're fine. But we are nowhere like that.
This is still on the same topic.
Yeah, apologies if it's sort of going over what we've already covered, but it's understanding this 6% because a lot of the people that have been in touch with me over the years looking for a puppy, for example, and they've lost it due to JKD, when they talk to me about what is behind on the pedigree, it is just a mishmash of non-prefix breeding, if you like. So it's kind of the studies that are going on. I'm concerned that it's not a complete picture of what is out there, because 9 times out of 10, the people that ring me and say, I've lost a puppy to JKD, and I say, well, what's behind it? You know, where did you get them from? And it's somebody that's just had a litter up the road because they felt like it. It's very, very rare somebody rings me and says, I've had a puppy from X kennel. And that worries me because, do you know what I mean? We're doing our absolute utmost and are concerned, and we want to put things right where we can. But there's all this other breeding going on that we have no control over, that is causing heartbreak for all these people on Facebook that find, you know, Molly's just died and her dad was called Red Rover and her mum was called Molly the whatever. Do you know what I mean? And it just bothers me really that the reflection of what is going on isn't a true reflection.
I think the last box of the last quarterly that came out with the breeding, I think I kind of worked out it's about 95% pet litters.
Yeah.
And it was only 5% that were actually breeders or known stud dogs that were used.
Yeah.
So I think it's a huge take-home message. I mean, I sympathise fantastically, but the trouble is, in order to do a study, you need the information, and I think that's why reporting these and encouraging people to report, and even if it's just a case, even if you just know it's, you know, just recording the sex ratios of, you know, the individual dog, anything about it is useful, because, you know, it's not a common disease, but it's so nasty that we want it wiped out, and we want. And to do that, we've got to understand where it's occurring, the conditions under which it's occurring.
So you think it can be wiped out?
Yes, it's not easy. If we could identify the gene and get a genetic test, and we simply say you should not breed from anything that carries this. I mean, there is going to be a problem with people with pet grade dogs who are breeding and don't.
There's no control over them.
There's no control over them. However, you know, one can encourage vets. I think the days of increased DNA testing as the cost per test goes down is going up, just like microchipping is becoming, you know, increasing. And you know, we could get to a stage whereby most pets are DNA tested, and
most of the pet people won't even get them heart tested, never mind, you know, it will be
No, I agree. I mean,
So
this
maybe
is
then
not
it has
an
to
easy
come
battle,
from then the vets then.
but I think,
Maybe that's where it has to
Yeah.
come from. That more that the vets have more knowledge, then they pass it on to the pet people.
yeah.
That's a good point.
Okay, folks,
It's been half for years to get this stuff done.
I'm going to
Yeah.
take another question because the time's been
Yeah, it follows on the coefficient thing really. I think this is for Bruce. Looking at the JKD website, the UK pedigrees section of it, I think it's 97, counted them last night. But of the 97, there's 36% of those 97 which have coefficients lower than 6.3, which is the Kennel Club's average for the breeder this year.
That's pretty low though.
I mean, the coefficient's getting lower, the average is getting lower, it has done over the last few years. But I'm just wondering why that should be. It doesn't, to me, it doesn't make sense.
But that's because there's so many pet people breeding that are breeding from willy-nilly dogs, isn't it?
Well, what are they doing differently? What are their vets giving them differently to what breeders are giving?
One sec. So that who's your question name?
Yeah, all I'm saying is,
there's over a third of the dogs on the JKD website, UK dogs, with a coefficient of less than 6%. And I was surprised when I saw the numbers. Most of those aren't pet dogs, most of those are well-known dogs. Some of them have got really high coefficients, but there's 36.8 is the percentage.
Yeah, I'd have to have a look at that myself, but general comment again. The pet population, really one can't do anything directly, because I get the pedigrees in, it doesn't make any sense to me. There's nothing there because there's no dogs being inbred, so you can see a pattern. There are no dogs used highly extensively like you get in the show world. The pet population is a real problem, and something can be done about it with gene testing when you get that. But as of now and as in the past, whether it be with cardiomyopathy or with PA, all you can do is to try and get the top breeders to do some selection then, because whatever they do filters down into the pet population. It does not come up. So you can't do anything about the pet people directly, but if they are getting stuff coming down from the show population all the time, if it's good for them. If it's bad for them. So that is one part of it. Then as to, I think we all know, who've been in boxes for a while, that many years ago, I think Philip made the point, you had many distinct lines of boxers, people favouring one type or another. There was a great diversity, whereas now we're pretty much all in a hole together. There's no clear way out. You can't go to a clear line, really, apart from going across. So how do you deal with this? But
maybe the kennel club need to help more then.
Everybody needs to help.
So I'm not a breeder, and I don't know an awful lot about the kennel club statistics or anything. But from a biological point of view, I mean, I think the observation that you make is very interesting, and I wonder if the reason for that is because actually the gene is quite prevalent in the population. So if actually you've got an awful lot of heterozygous dogs in the population, actually you are going to get affected animals, even though they are relatively outbred. We tend to think of genes as and mutations as being quite infrequent, but actually that's not always the case. So if you take the example of the Dalmatians, Dalmatians, as you probably know, get urate stones, so they're stone formers, and actually until some new dogs were imported, all purebred Dalmatians have two abnormal copies of the gene, so they all produce too much urate. So those Dalmatians, if you looked at them actually, and you looked at their inbreeding coefficient, it wouldn't relate to whether those dogs got stones or not, because there are lots and lots of copies of that 40 gene in the population. So what it may be is that actually this disease has always been there in the boxes, and that there's a lot of heterozygotes out there, and that's why we're going to need a gene test, because it's only going to be with a gene test that can pick up the heterozygotes that we're going to be able to stop it happening.
Nigel, I've got a question from the live feed.
Okay.
Somebody's asked, can a whole litter of 8 puppies have JKD? I imagine I can probably answer. Says have, so I imagine have the symptoms.
Could they? They could if you had 2 affected. So if the mother and the father were affected and you bred them together, then all the offspring would be genetically affected. I think if you have a whole litter with kidney disease, then you'd have to look at other potential causes as well. So some common toxin or infectious thing or something like that, and then that becomes very likely. But genetically, it would be possible if both the dam and the sire were affected.
Okay, thank you.
Now, I've been asked this question many times with whites, and of course you can get whole litters of whites.
Yeah.
But then you only find with JKD only a proportion of them. So the chances of getting a whole litter affected is not very good.
The amount of kidney
Gonna do
needs to be
Philip
affected.
and then Robert, okay?
It's just something I'd like to share with you. My wife and I, we do rescue in South Wales. The lady back there was talking about a pet bred litter, and we had some couple of years ago an incident where a lady approached us, her puppy was ill, and my wife actually spoke to her, and what was determined in the end was nearly unbelievable. The people who had bred that litter had sent these people away with a 6-week-old puppy, a loaf of bread and Marmite. What, it wasn't a laugh, no laugh. Most of that litter died from salt. It does kind of bring me to the bit where you were talking about food again. Nowadays, dry food is very popular. If dry food is fed without the support of adequate water, a similar occurrence could easily occur as I've just described with the Marmite, I believe. Could you, but I was just wanted to tell you, it's when people who are not involved in the breed get involved, they do not read the necessary work and they commit all sorts of unbelievable sins. And that could just as easily be what happened with this lady. Sorry.
I haven't specifically studied that myself in dogs. I have actually vets tend to say if a patient, or vets tend to say, oh, you know, feeding dry food might predispose to kidney disease. So the main area of my research is actually on cat kidney disease, which is very common. If any of you have cats, you'll know that's very common in the older cat population. And we did do studies are never big enough, but we did do an epidemiological study where we looked to see if there was any association with feeding moist or feeding dry food, and we couldn't find an association. We haven't done the same.
How big was that
study in dogs? Not huge. So we had I think we had about 100 cats with chronic kidney disease and more that didn't have kidney disease at a really reasonable age. No, these were cats.
Just cats.
And there was and we had diet histories from them all, and we looked back at that.
So it'd be interesting to do it on dogs.
It would be interesting, but probably more interesting for dog kidney disease across the board rather than this problem in particular. But I think in all breeds, looking at the risk of it, but there hasn't been any proven association with that so far.
But that's just with cats, though.
My work has just been in cats.
Right,
I'm
okay,
trying to
that's
think
fine.
whether there's been a study in dogs, and I'm not sure whether
So it
it's
has to be
been
then.
done.
So it has to be, surely.
What about
A study?
What about doing a study in vaccinated and unvaccinated dogs?
Well, I mean, I think the populations of unvaccinated dogs that you have are so that there's a really strong confounder effect there because unvaccinated dogs tends to, come from shelter and have poor healthcare and other problems. Although now, of course, there is a move towards people not vaccinating, but I'm not going to do any study which advocates not vaccinating dogs.
I'm going to move on to Robert. So, Robert, you're next.
Sorry, but I've got Robert Vanson. Sorry, I've got 3 questions. Thank you to Bruce for offering to put out your slides. Would the other 2 speakers be prepared to do the same? Make your slides available for us?
I'm happy for my slides to be made available with the caveat that some of the pictures were stolen from the internet. So I should really do that.
You can redact those if you wish.
That was just to make them prettier. I mean.
Let's say we are recording, so that should be available on
I'm going
Facebook,
to put it on
wherever.
the Facebook.
The true questions are, Professor Meyer was on about the size of kidneys and whether vets can identify if it's JKD or something else. Does the size of the kidney development, does that indicate any form of JKD, or is it just any kidney disease through lack of development?
Do your next question before.
All right, okay. And the other one was specifically for Bruce, which was how far back have you identified JKD producers in pedigrees?
Well, I think the Leslie Boyle would be my first experience with going in, finding out years later. But 1980, I'm sure it's an American dogs and continental dogs which have a very different history, as you know, from the development of the breed. So I expect JKD has been around since pretty much the beginning of boxers, but it is allowed to continue because most of it's hidden.
Yeah.
You can make 2 carriers together again and again and again, and you might find difficulty finding any because most of them are not found for whatever reason. So it is an insidious disease. I mean, where, I talked about you've got a bunch that you recognize, and there are missing ones, and they are here. Now you find 10% are affected, the other 15% are up here, So you're thinking there's not very many, we don't have a big problem, but the smaller the cases go down, the bigger the risk of double those animals being around and bred from. This is a nasty thing about it, you can keep it going for ages. I mean, I mentioned Sheila Cartwright. She has obviously seen things around for a very long time, going back for the whole of my history in boxers at least. So I don't think there's something new, but we have zeroed in. It's all got narrower and narrower and narrower, partly because of other things we've dealt with. I mean, cardiomyopathy really got hit far too hard. People wouldn't touch anything that was at all related to cardiomyopathy. We lost stuff. Shouldn't have done. It's too hard, which is why I put the slide up there. Let's not go too hard. Not that you can now, because everything's got pretty much the same pedigree. But it is interesting to look at some of the current show catalogues. It looks to me, as now an outsider, that the breeding has changed dramatically over the last couple of years. I don't recognise the dogs. It's something
Robert, the topic of your second question was?
The first one was the difference in size of kidneys.
Kidney size.
Yeah, so seeing that the kidneys are smaller and things, it tells you that they have kidney disease. It doesn't tell you that it's JKD specifically. And I don't even know, unfortunately, whether that is the typical appearance of JKD, because so few of them have been well described. So they could be small, large, lumpy, bumpy. The only descriptions are in the Chandler study, and that they just look like diseased kidneys, but very variable in their appearance.
Thank you. Other question from the floor? Thank you.
Hi, it's Catherine Thompson. I just wanted to ask, obviously we've came here today to learn and understand more. When we leave here today, what can we do to help?
Well, as far as I'm concerned, I think there needs to be more research done, mainly to find where these missing dogs are. Because if they're dying baby puppies, it's not bad for the breed, it's just a waste. But if they're breeding and going on, we have a major problem. And the only real way we can get out of that, apart from selective breeding, is to get a gene test. So it's research, and frankly, as Bill mentioned, any information that can be collated, understood, whether it just be the sex of the dogs, anything would be useful. But there's got to be more communication, feedback.
I'm still collecting samples, particularly from affected dogs, and litter mates of affected dogs will be particularly useful. So if you've got an affected and an unaffected dog, so you bought it from one place and you can go back and track it down, then that's particularly useful. I've got a big sample set, but as everybody's been discussing, this is not a simple disease, and I'm afraid it's the one place when I rather disagree with Bruce that it's a one gene recessive. If it was that simple, I think it would have popped up by now. There are clearly other things going on. There's something causing the sex ratio distortion. There probably uneven penetrance, which might explain part of that. But the more complicated the genetics, the bigger the sample size you need in order to track it down and find out exactly what's going on. So yeah, more samples from affected dogs in particular.
When you say samples, are we talking about swabs?
Swabs, yes. Swabs.
Is that the best samples that can be provided?
I mean, if a dog dies, then actually a small piece of skin is fantastic because that produces essentially unlimited DNA for future studies. What I've been trying to do is to encourage anybody who sends samples. I mean, it's horrible. You've got somebody with a dog that's about to die, and you don't want to say, please can I have lots of bits. But pieces of skin, if you're prepared to donate a piece of skin, it lasts essentially from forever, and it could go on to other studies. You know, I'm not going to be researching this forever, but I hope that my panel of samples, you know, it's already 1,200, it is growing, and it does mean that, for example, they're interested in Scandinavia and they're doing studies, and it may be that we can share data and make it even bigger. But certainly the swabs absolutely fine for you know, that allows me to do 100 tests, which is, you know, fine. But for future stuff, it's good to have a bit more in the bank.
Could I just jump in there before? Sorry, Professor. You mentioned about skin samples.
Yes.
I was once told by a researcher that vets tend to destroy the samples through the way they store them. Is this correct? Is there a particular way you want the...
So DNA is fantastically tolerant, so skin can be, worst case scenario, you can just cut a very thin slice and dry it. Dried material, you know, they get DNA from mummies. If you're desperate, putting table salt. I used to work on marine mammals, so we'd have somebody says, I found a rotting lump on a beach, do you want a sample? What have you got to hand to preserve it? Table salt is good, but usually if you contact me, I can send tubes with non-toxic preservatives, which will be fine for posting, whatever.
And I was just going to say, in terms of if there is an animal that you know of that is diseased, or actually, even if it hasn't got kidney disease, if it's dying for some other cause. I'm not sure if Jessica will take the unaffected ones, but any dog with kidney disease, trying to get a sample of their kidney to her. And if it's a patient that is dying, then often the vet will there won't be the same charges associated with collecting a biopsy if the patient's already dead. And that's where what the vet will do if they don't know that you want to collect a sample will destroy the tissue, because what vets usually do is to freeze the deceased patient before they go for cremation, and freezing the tissue destroys it so that they can't look at it. Now, the limitation for what Jessica wants to do in an ideal world, and I don't know, she may be struggling to collect cases and she may accept material that is non-ideal, but in the ideal world, a bit of kidney is put in a special fixative, which allows her to actually look at it under the electron microscope, and your vet will not have that special fixative on their shelf. If there's a patient that is declining, contacting her and she will get some of that special fixative to the practice for when the time comes. But I think getting her kidney and tissue, it doesn't find the gene, but it gets us closer to understanding whether there are a multitude of problems or how they manifest. And actually the two things in concert, sort of understanding, okay, the ones with the gene looks like this and the ones without the gene, that's going to be ultimately the way to sort it out.
Yeah, just out of interest, how can it be quite a while after death, or does it have to be within 7 hours? Or does it have to be whilst they're still circulatory?
It can be taken shortly after death, but kidneys unfortunately degenerate really quite quickly. Certainly we, for my cat research, we go to our clinic, and then if we put a patient to sleep whilst we're at the clinic, because we run a charity clinic, then we go back to the main hospital. So a few hours is okay, but days and days is not.
So do I understand you'd be happy to look at kidneys from older dogs that are suggested? Or is the numbers getting too high? So obviously we'll be interested in the ones which are normal, not the ones which are affected.
Yeah, so this is me plugging somebody else's study entirely. I have nothing to do with the study, and so I don't know. I know she's got funding from the American Kennel Club, but I don't know how many animals she's got funding for, or when she will say, stop, I've got as many as I can afford to do. But for so I'm sure it's a finite number.
The
but
costs are high, or
so doing the renal histopathology, including the electron microscopy, and having there's a nephropathologist at Ohio. So that's somebody who is trained as a veterinary pathologist at looking at tissues, and has done years of specialties training in that, but then she also went into human medicine and got a human specialist qualification in looking at kidneys, because what is understood and the recognition of diseases is so much more advanced in human medicine. So she did the whole sort of human training, and then she came back to the veterinary field. So her assessment, when it's offered commercially sort of across the board, it's $850 just for looking at the tissues, but that is provided free of charge if it's from a boxer. So it's a pretty good deal.
And I would add, just to emphasise the fact that, I mean, it's a shame there's only this one person, so apparently, but actually the morphology of the kidneys is not, I have no expertise at all, but the more we define the phenotype, if this really is what we call JKD, is actually 2 or 3 diseases that are related but have slightly different causes, that's so important to find out. I mean, that's, in a way, the earlier one can find that out, the better, because otherwise it just essentially muddies the whole water.
That's call time at 4 o'clock. I think that's reasonable, isn't it? Any more questions?
This is, I posted on Facebook earlier, so people just sent me questions that I had that I could ask. This one is: the entire JKD debate seemed to gather steam after the BBC programme, which seemed to lay prominent blame on a prominent stud dog. And much was implicated about the kennels and their use of such a dog when they supposedly knew the dog produced JKD. And this type of attitude is now rife among the show world whenever a pedigree shows up on the published list. Does the team regret putting forward such accusations towards various kennels before all the facts are in, as it seems to have done much more harm than good getting the much needed support from all the breeders? Side hundreds of puppies, by comparison only cite a few cases. Therefore, seeing as there is no concrete understanding of inheritance, it is not possible that he is not solely responsible. And it could more than likely been carried through the bitches involved, not only through the inheritance, but external forces such as E. coli or vaccine reaction. And were all the cases he was involved with, or diagnosed, were they diagnosed through autopsy? I think it would be very helpful to have clarity over this prominent case. That was sent to me.
Well, I mean, this is really kind of crazy, because, I mean, every one of you will have seen the pedigrees, and I don't think any of you would have doubted that you could see the picture is there. As to, what was the last bit? The last end?
Side hundreds of puppies.
Yes,
Well,
the more puppies you sire, the more you find. But he did have a lot of puppies, a lot of dogs have had puppies. He sired more than any other dog. Inbreeding on him is continuing to produce more and more cases. And it is only now that breeding seems to have changed a bit that maybe things are going down. Breeders will find a way out. Now, is it justified? If it's not justified, I got to go shoot myself, because frankly, that is as hard, nailed down as hard as anything I have ever done in dogs, or indeed my scientific career. There is no doubt at all that JKD is inherited, and it's got to be quite simple in that you can get dogs from all over the world showing the same pattern of inheritance and combine together to give the same thing. You just cannot imagine any scenario which could be much more complicated than that, other than the difficulties we've talked about already. You know, if you don't find enough cases, they're there somewhere, and if they're not obvious, you know, affected animals, they are the ones you can't see, and that's even worse. Though you don't see them, they are spreading it around.
Anybody, do you want to respond to that?
We had one more question. Has the mitochondrial DNA been looked at?
Not by me.
Okay.
It should be maternally inherited if it's mitochondrial, shouldn't it?
Yeah.
I would have thought.
Yes.
So
say that again, please.
So if it was inherited through mitochondrial DNA, it should only be inherited through the maternal line.
Yes.
So it would be a different pattern of inheritance.
but if it has low penetrance, it will occur, obviously, a mother produces both sons and daughters, so although it couldn't be the son who passes it on, you could still get an incidence with complicated genetics, additional genetic stroke environmental factors that could create a misleading pattern. So I think it's very unlikely, but you have to consider everything.
Yes.
So.
And you could get every dog in the litter could be affected if it was maternal.
Yes.
Every single one, if it was mitochondrial.
Yes.
Okay, folks, any further questions? Okay, I'm going to ask John to bring proceedings to a close.
Well, I think we should all thank our speakers for such interesting talks. I mean, I've had a terrific afternoon. I've really learned a lot, and we think it's been a very lively discussion. And so I'd like you all to join with me in thanking the speakers, and also thanking Nigel, who stepped in at the last minute to chair the session because Rick Beale couldn't be here. So, okay.